Hey guys, Dr. B. This is just a short video for an exam review to give you an idea of what you're going to see on the test Wednesday. I'm going to kind of go over some major parts of the exam study guide, then I'm going to show you some questions from the actual test. The test will be paper, but the ones I'm going to show you are an electronic version of it, but literally these will be questions that are actually directly and actually from the test. So basically how this is going to work, you have a bunch of multiple choice questions, and then there's a few that are open-ended, and the ones that are open-ended are the ones that are in red. Pardon me. So everything in red collectively is worth, maybe 30-ish something percent of the whole test, maybe up to 40. I don't remember exactly how much, but it's in between 30 and 40 percent. So let me just kind of give you a sense here of kind of how deep, because obviously the question a lot of times is, well, how much do I need to know about a topic? When I give you a study guide as specific as this one, and meaning this is literally what's on the test, the answer could be literally as much as you can possibly find out. So I'm going to go through and tell you the ones that you really want to go deep with, and some of the ones that it's more of just, know its definition or whatever. So in terms of the top three, the first three right here, you want to know them in pretty good detail, even a few examples of media as well. As well as microscopes, you want to be able to tell me the fundamentals of the different types of, like for example, light microscope, dark field, electrons, stuff like that. The rest of this is fairly straightforward down the line here. So knowing the different staining techniques in general and why you would use them. But you'll see here that out of one of the stains, there's a lot I want you to know, and that's the gram stain. And we're going to do that in lab probably next week. Also, how to isolate a single colony of bacteria. That goes right along with the 6 I's. So you want to be able to understand how to do that. One of the more declarative knowledge, which really means just memorize, not to say memorize, but just really terms. There's no other way to get around it. These two would be definitely terms, knowing what the major parts are. And you've had a lot of this before in BIO250, so that's good. Maybe not the flagellar arrangements, but definitely the bacterial parts. Moving down the list, endospores, you really just want to fundamentally know what their purpose is. Like, why would bacteria create an endospore? And I'll give you a hint. It's not for reproduction. It's way more for protection, but you can talk about it later. Understanding what the archaeobacteria are, in the sense that those are not the ones that are common. They'd be ones that found in like Antarctica or the bottom of the ocean in thermal vents. They're way more unusual than the rest of the mesophilics. So relative sizes of different types of microorganisms, understanding, like, for example, a human cell to a yeast to a bacteria to a virus, like who's who on the size chart would be what I mean by that. Obviously, any of the files, understanding what they might mean. And then down here, these two, really more fundamentals, understanding what a protist is and what makes a protist, and what an algae is, and its characteristics. I do not need you to go through all the different phyla of algae and protists. That's ridiculously boring, and you could always look it up on your phone. So it's not about memorizing the different types of algae, but it's understanding what makes an algae and what makes a protist, which leads me to characteristics of molds or fungi. What are they, and then what do they do? If you were to interview a fungi, hey man, you're a fungi, what's up? What would it say? Hey, I do this, I live here, I eat this, I get rid of that. I don't like this, I do like that. That's really kind of where we're going. So obviously characteristics of helminths, not much on that except generally what they are. So maybe a question or two on helminths, not much else. Eukaryotic cell organelles, again, just knowing kind of what they are, what they do. That's more of a listy thing. Again, the obtaining pure culture from a mixed goes way back once again to the 6 I's of micro. This couldn't be any more straightforward than the way it's asked. Explain how a biofilm differs from a typical colony. I wouldn't be surprised if that's literally directly the same verbiage. And then finally, again, the characteristics of how these things might be the same and might be different. So that should give you a little more of a clue. So let me show you now 3 or 4 questions just to give you an idea of kind of what you're going to see on the exam. Now, this is not necessarily number 4, so don't worry about that, but this question is directly on there. I believe you may have had one like it on the quiz, but the reason I'm showing you this, and again, I'm not going to tell you the answer because I'm showing you the question, so go figure it out. But you can see here that a microbiologist inoculates a plate with 2 different organisms, and then after they're incubated, only one of them grows. Basically, why? So I could ask, the reason I showed you this is I could ask it to you this way, or I could say a microbiologist inoculates 1 organism and 2 of them grow. Why would that happen? So that's a perfect example of something you'd see in terms of pure cultures versus mixed cultures versus media, etc. Here's a perfect example of the level of questioning for like the microscopes. In general, I just need you to know kind of how they differ from each other. And obviously, we'll be spending most of the time on the bright field or the compound microscope in terms of magnification. But this is just more of a general question about generally the difference between them. Here's a good question, just kind of showing you, an understanding of how bacteria would differ from, for example, a eukaryotic cell. That's why I like the question. So it's, what are these, which one of these would a bacteria not have? And so that's a good way to ask that, to get beyond just the memorization part. And again, here's a question as well, more so just kind of getting into the characteristics of fungi. And again, obviously I don't need to tell you the answer because, well, the green arrow tells you what the answer is. So, these are just a couple examples of the ones that are multiple choice to kind of give you an idea of the level of questioning. I'm now going to show you one of the open-ended questions just so you get a sense of what that's like. This is a super important question. It's absolutely why I'm going to show it to you. It's not a secret. It's literally on the exam study guide. But I definitely want you to put some time into this. This is kind of what the entire first part of this first unit's about. There's a contaminated food sample. Microbiologist wants to study one of them. How would a microbiologist do that to get a pure culture of just the one? So, obviously we're talking about the 6 eyes, but, be careful. They don't necessarily go in order. It's used in terms of, their application. So, a lot of times you're inoculating on a plate that is already, in essence, if you're using a selective media, done some of the isolation. There's a bunch of ways to answer this. There's a lot of right answers. But just convince me that if I were to follow that protocol, I would know how to end up with what the question asks. So there we go. There's your exam review. If you have any questions, let me know, and I'll see you guys Wednesday.